Once again, welcome to ReviseMed YouTube channel for pharmacology. Today, we are going to see something about source of drugs. Along with that, we will also see some aspects of drug development. A short notes may be asked from source of drugs. It may be asked natural sources. Sometimes they may also ask us synthetic source of drug development. Whatever it is, you will be able to write the short notes if you follow this particular episode. Sources of drugs and drug development. The earliest medicinals were pure natural products. They were crude powders, juices, or extracts from animal, plant, and mineral sources. These continued to be the only source of drugs till the early 19th century. Folklore and chance observations also led to several discoveries in the early periods of drug development. Morphine for pain relief, cocaine the first local anesthetic like alkaloids, and digitalis like cardiotonic glycosides are still used in medicine came through this route. From plants we got alkaloids like morphine, glycosides like digitalis, tannins, oils and resins. Of the mineral sources which we get from earth and soil, iron, sulfur, potassium and even gold are important drugs. From animal sources, we get insulin, growth hormone like products from the glands, organs and tissues of animals. But there are several limitations to the natural sources. They are limited possibilities, prepared by individuals on small scale, limited administration, not purified, not standardized or tested, no controls to test their efficacy, no idea of mechanism of action. Now coming to synthetic drugs, drug invention became more allied with synthetic organic chemistry as that discipline flourished over the past 150 years. Paul Ehrlich's work with dyes culminated in the invention of arsphenamine in 1907, which was patented as salvarsan used in syphilis until the discovery of penicillin. Gerard Domac demonstrated that another dye, prontosil, the first clinically useful sulfonamide, was dramatically effective in treating streptococcal infections. This launched the era of antimicrobial chemotherapy. Then what are the other sources of drug development, or what are the other sources of drugs? From microorganisms, we are getting all sorts of antibiotics like penicillins, cephalosporins, tetracyclines, etc. And we also get antifungals from microorganisms. Of human source, we get immunoglobulins, growth hormones, chorionic gonadotrophins like drugs. Genetic engineering has revolutionized drug development and also the source of drugs. Cell culture gives us urokinase. Recombinant DNA technology gives us human insulin and its varieties. And from hybridoma technique, we are getting monoclonal antibodies. What are the advantages of synthesis and other recent methods? Unlimited possibilities, prepared by very large companies on massive scale. They are highly purified, they are standardized and tested, they are available worldwide, tight legislative control and mechanisms partly understood. There is a lot of change in trends of drug development. Now they are telling more of inventions than discoveries. The era of discovery has ceased. Now the era of invention has started. So for those students who would like to know something about current trends of drug development, this particular message will be of some help. Now for students who would like to know some more detail about current trends in drug development, I would like to share the following information. Today, useful drugs are rarely discovered. They are invented. Invention emphasizes the process by which drugs are sculpted for specific condition and brought into clinical use. First, one way is to synthesize close chemical relatives of a drug known to inhibit some enzymatic activity. So, this new chemical which is known to inhibit the enzymatic activity, may in future become an anti-cancer drug. So, several recently discovered or invented anti-cancer drugs are of this origin. You will wonder what is druggability. We can modify the structure of a drug. Now we are in a position to modify the structure of a tissue or receptor or target organ. So, by genetic engineering, we are able to modify even target receptors to accept a drug we have designed. Then proteins can be designed, customized and optimized using genetic engineering techniques. Proteins used therapeutically include hormones, growth factors including erythropoietin, granulocyte colony stimulating factor, cytokines and a number of monoclonal antibodies. Now, everybody knows the uses of computer. So, drug development is also using computer. Computer-aided drug development. There is a trend in drug invention to achieve success entirely through computation. On one side, imagine a database containing detailed chemical information about millions of chemicals. On the other side, a second database containing detailed structural information about all human proteins, say drug targets or receptors. So, we try to fit the chemical with the protein and find out whether the drug acts according to our expectation. With the computational approaches, we are able to find new uses for old drugs. Thank you.